Good morning, everyone. Today, I'm going to discuss about an important topic, one of the malabsorption syndrome, that is called celiac disease. What if the food you eat every day slowly destroys your own intestine? How it sounds. So let's understand what celiac disease is. First, understand the definition of celiac disease. See, celiac disease is also known as celiac sprue, or it is also known as gluten sensitive enteropathy. It is an immune mediated disorder. It is an immune mediated disorder triggered by the ingestion of gluten containing foods such as wheat, rye, or barley in genetically predisposed individuals. Basically, our body is developing an abnormal immune response against gluten containing foods, that is wheat, rye, or barley, in particularly in genetically susceptible individuals. Before understanding the pathogenesis of celiac disease, it is very important to understand what gluten is. What is gluten? See, gluten is a major storage protein that are deposited in the starchy endosperm cells of the developing grains, and it has viscoelastic properties. And this gluten is made up of two components, one is gliadin and another one is glutenin. This gliadin is an alcohol soluble fraction of gluten, and it gives plasticity and elasticity. It gives plasticity and elasticity, and gliadin contains many disease producing components. Okay, gliadin contains most of the disease producing components, and this glutenin is a water soluble fraction of gluten, and it provides strength and structure. So this gliadin is the one which is responsible for the disease component of celiac disease. Next, we will understand what are the environmental factors and risk factors for celiac disease. Environmental factors. So, environmental factors are the important determinants in the development of celiac disease in genetically susceptible hosts. Then, who are these genetically susceptible hosts? Then, who are these genetically susceptible hosts? Those are the individuals who are carrying the genes for HLA-DQ2 or HLA-DQ8. Those individuals who are carrying the genes for HLA-DQ2 or HLA-DQ8 are genetically susceptible individuals in case of celiac disease. And what are the proposed risk factors of celiac disease? Those are infections including rotavirus or reovirus, low birth weight, as well as antibiotic use. These are the proposed risk factors involved in the development of celiac disease. And there are some non-HLA genes which are also implicated in the development of celiac disease. These are involved in regulation of T cell differentiation, regulates T cell survival, regulates T cell migration, and regulates T cell cytotoxicity. Regulates T cell cytotoxicity. Now, we'll understand the pathogenesis of celiac disease at cellular level, and the entire pathogenesis is depicted in this small picture. I had mentioned that environmental factors play a major role in the development of celiac disease. Whenever the cell is under stress, because of any of the triggering factors which I have mentioned earlier, the cell which is under stress expresses an antigen called MICA, which is depicted in the picture also. MICA is basically MSC class 1 chain related protein. Now let's see what happens when a person consumes gluten in the diet. Normally, gluten is digested in the lumen of the intestine by various enzymes. One of the component which is released after digestion of gluten is gliadin. When gliadin peptide interacts with intestinal epithelial cells, you can observe in the picture. So gluten is digested and now the gliadin peptide is released, and when this gliadin peptide interacts with intestinal epithelial cells, when gliadin peptide interacts with intestinal epithelial cells, it stimulates intestinal epithelial cells to release interleukin 15. See, when this gliadin peptide interacts with intestinal epithelial cells, it in turn stimulates intestinal epithelial cells to produce interleukin 15. Then what is the role of this interleukin 15? This interleukin 15 causes activation as well as proliferation of intraepithelial lymphocytes. See, you can see in that picture T. between two epithelial cells, intraepithelial lymphocyte is present, that is represented by T in the picture. Once this intraepithelial lymphocyte is activated, it expresses a marker called NKG2D. So once these intraepithelial cells are activated and proliferated, which is stimulated by interleukin 15, they are going to express a marker called NKG2D, that is called natural killer group 2D receptor. That is called natural killer group 2D receptor. Actually, this NKG2D is a receptor of MICA antigen, which is expressed on the cells which are under stress. Therefore, these intraepithelial lymphocytes which have expressed NKG2D will now attack the cells which have expressed MICA antigen on them. So now cells with MICA are attacked by NKG2D expressing lymphocytes, causing epithelial damage. If many such epithelial cells are damaged due to interaction between intraepithelial lymphocytes and epithelial cells, now gliadin peptide can easily enter into lamina propria. In the lamina propria, this gliadin is deamidated by one enzyme called tissue transglutaminase. So because of damage of many epithelial cells, the gliadin peptide can easily enter into lamina propria, and this gliadin is deamidated by tissue transglutaminase 2, resulting in the formation of deamidated gliadin. Now this deamidated gliadin interacts with antigen presenting cells in the lamina propria, and this antigen presenting cells has a receptor for deamidated gliadin, that is HLA-DQ2 or HLA-DQ8. Once this interaction happens between deamidated gliadin and the antigen presenting cells in lamina propria, which are having receptors for HLA-DQ2 or DQ8, stimulates CD4 plus T lymphocytes, and these stimulated CD4 plus T lymphocytes will produce interferon gamma and further damages the epithelial cells. And now this stimulated CD4 plus T lymphocytes also stimulates B cells to produce several antibodies called as anti-gliadin antibodies, anti-endomysium antibodies, and tissue transglutaminase antibodies. So this is the entire pathogenesis of celiac disease at cellular level, and the same is summarized in that flow chart. Now we'll understand the symptoms of celiac disease. Gastrointestinal symptoms, most prominent presentation in celiac disease, like diarrhea, chronic diarrhea, constipation, most common in children. There can be abdominal pain or discomfort. Due to gas production, there can be bloating or distension, delayed gastric emptying or heartburn. Then steatorrhea can be there, weight loss in severe cases, failure to thrive or growth retardation in children, and a nutritional deficiency because of malabsorption. Why malabsorption is present? Because of destruction of the villi of the proximal small intestine by those antibodies which are produced. Iron deficiency anemia, then vitamin D, vitamin B12, folic acid and zinc deficiency can occur due to malabsorption. Vitamin A, E or K deficiency can occur if steatorrhea is prominent. Then certain skin manifestations, some individuals can develop itchy, blistering skin lesions, also called dermatitis herpetiformis. So this is about the symptoms, signs, symptoms, pathogenesis of celiac disease at cellular level, environmental risk factors, and what is the role of non-HLA genes, and what is gluten, and the definition of celiac disease. In the next video, let's see how we actually diagnose the disease through investigations and biopsy findings. For such more clinical topic discussion, like, share, and subscribe the channel. Thank you.